Welcome to PXE International's webinar with Dr. Váradi. Again, I'm going to just give you a few housekeeping details before we start. You are all on mute, so you can't ask questions verbally, but you can write them to us, and we will present them to Dr. Váradi in a question and answer session at the end of the webinar. To do that, you go to the question pane, type in your question. The question pane is in the control panel on the right side of your screen. And type in the question, and we'll keep them until the end of the webinar and then present them to Dr. Váradi. Also, if you have any technical issues, type your question about that into that question pane too. But first, try to log out and log back in. That usually fixes most of the issues. So, welcome everyone to the webinar, What is the PXE-Associated ABCC6 protein doing or not doing in the liver, and can we fix it? I'm delighted today to welcome another great researcher of PXE and a friend of PXE International, Dr. András Váradi. He joins us from his office at the Hungarian Academy of Sciences in the Institute of Enzymology in Budapest, Hungary. He is the head of the Laboratory of Active Transport Proteins there. He founded that laboratory and his own research group in 1989. It is devoted to the study of the molecular mechanism of active transport proteins. In 2001, Dr. Váradi started to study the function of the ABCC6 protein, which is associated with PXE. His lab was the first to express the protein, demonstrate that it had transport activity, and show that loss of that transport activity is the cause of PXE. András has been a plenary speaker on ABCC6 proteins at a number of important meetings, including the special meeting on ABC proteins in Innsbruck, Austria, and Gordon Research Conference meetings on elastin and elastin fibers. In 2007, his laboratory became a founding member of the Euro-PXE Consortium, a European PXE research network. And in 2009, Dr. Váradi, together with PXE International, Sharon and Patrick Terry, and Dr. Guido, organized a satellite symposium on PXE at the ESDR annual meeting in Budapest, Hungary. He has published over 50 papers on ABC transporters. He also teaches human molecular genetics and molecular biology at 2 universities in Budapest and supervises the projects of graduate students, some of whom research PXE. Today he will be presenting some of his research on PXE in a webinar titled, What is the PXE-Associated protein doing or not doing in the liver, and can we fix it? Dr. Váradi, welcome, and thank you for being with us, and I'll hand it over to you.
Thank you very much. First of all, can you hear me well?
Yes, I can hear you.
Great. So I should like to thank for the opportunity to be able to discuss our research with the audience, and I hope that at the end you will be as excited as we are doing this research. So first of all, I'm a protein chemist or protein biochemist by background, and therefore mostly, as the title says, I will talk about the protein and a little bit about the liver, and probably a little bit less about the disease. But let me start with an introductory slide which shows that what is the basic pathology of PXE. This is generally accepted that PXE is somehow a member of soft tissue calcification disorders, and this soft tissue calcification, which can manifest in the skin, in certain parts of the eye, in the arteries, and all over in the body, can be due to several different things like aging, diabetes, and different other disease conditions. Now there is one group of the diseases which are genetically which depends on genetic conditions. This is what I would call ABCC6 dependent calcification, since those diseases, and PXE is the most important one, are due to loss of function mutations in the ABCC6 gene. As you may know, as I told you, and the whole seminar is about PXE, which is the most important ABCC6 connected or associated disease, but just for the sake of completeness, I want to mentioned that another genetic disease which is much rarer than PXE, the name is written down on my slide, I hope you can read it. About a year ago, that was also connected to mutations in ABCC6. But today, I'm talking only about PXE. And we consider, and there is a general agreement, that PXE is a metabolic disease. So why do we think that? The protein whose mutations cause the disease are present mostly in the liver. In biology, we call it expressed in the liver cells, but as you know, and I also mentioned, the symptoms affect the eye, the blood vessels, the skin, so they are extrahepatic, not in the liver. And the most plausible, most simple explanation for that we have here a transport protein, and ABCC6, as we showed that some 10 years ago, indeed a transporter, transports something, we unfortunately do not know what, from the liver to the circulation, and this unknown metabolite goes into all over into our body, to the eye, to the skin, and so on, and slows down calcification. Now, if this transport activity is damaged or lost due to genetic mutations in the gene, PXE develops. So the next slide shows a representation of the protein. This is a large protein and it's associated, or most precisely embedded, to the cell membrane. I show you now the cell membrane, and this territory is inside the cell. We call it intracellular domain or intracellular compartment, and this is extracellular. What you can see that the protein is represented as a long chain, and it is indeed a long chain, and this long chain goes in, out, in, and out through the membrane, actually 17 times. This is a very complex structure and keeps the protein very, very tightly connected to the membrane of the cell. On the upper right corner, there is a microscopic image of liver cells. This is one liver cell, and there is a special technique to stain specifically ABCC6 protein, which is indicated here with green color. The blue shows the nucleus of the cell. So you can see that the protein is always in the plasma membrane which surrounds the cell. And the idea is that it pumps something out from the cell into the circulation. So we discussed that it is mostly present in the liver, localized in the liver cells in the plasma membrane, as I showed you here. And what we also know that there are large number of mutations, and we will focus on one class of mutations called missense mutations, and those mutations are causative in PXE. Actually, they were identified by several groups all over the world in PXE families. Now, I have to explain you briefly what a missense mutation means, because in the rest of my talk I will concentrate on missense mutations. A genetic alteration can affect a gene on different ways. It can be that the gene is completely lost. Then there will be no protein, no function. It also can happen that only one part of the gene, and consequently the protein, is deleted, is removed, and then we will have a crippled protein which won't be functional, will not work. But most often we have smaller changes. A protein like ABCC6, but this is true for each protein, build up for smaller building blocks called amino acids. Our protein is much larger than average. This consists of 1,503 amino acids from 1 to the 1,503 number. So these amino acids are basically different, but the sequence and the position of amino acids in this protein chain are very well defined. It could happen that a mutation do not remove part of the protein or do not remove the whole gene, which results in no protein at all, but simply makes a little minute change in the DNA structure, which yields an amino acid replacement in one given position, like here, what we illustrated with red, dark red dots. That means that in this certain position, normally one type of amino acid should be present, let's call it A, but because of the mutation it's replaced by B. And this very often causes this type of mutation and this type of alteration of the protein structure very often cause problems. There are many missense mutations discovered, and I would think that more than half of the patients suffering PXE or affected by PXE carry one of these several hundred different missense mutations in the gene. Therefore, at one position, their protein is different than the normal, which in genetics called Y type. So the aim of the research we embarked on a couple of years ago are several-fold. First, we wanted to determine what are the consequences of disease-causing missense mutations on the protein. So again, I want to remind you that only one element of the 1,503 is changed for another one, and that can cause problem. So we want to figure out what is the problem, what finally causes the disease. So at the next step, we decided to look for mutations where the transport function of the protein is still preserved, but they cannot go to the plasma membrane. You remember that 2 or 3 slides ago, I showed you that normally the protein should be in the plasma membrane. If it's not in the plasma membrane, it cannot fulfill its function, cannot fulfill its hypothetical transport function, transporting something out from the cell. So if a protein is active as a transporter, but it's not at the right place in the cell, it's still a big problem, and it still can be the cause of the disease phenotype. So if we identify mutations with these specific features like preserved transport activity but challenge cellular localization, we can think about how can we correct the cellular localization, how can we fix this problem in vivo in liver. And if we are successful, we can come up with a suggestion that that given mutation, or the patients with that given mutation, or with those given mutations, we hope that there will be more than one found, can be subject of a specific therapy, which is in genetics called allele-specific therapy, means that depends on which mutation a patient has, this therapy either a possibility or not. So this is the logic of our research. This is the itinerary we want to go through, and that's what I want you to come with me and look up how we develop this project. So here is the protein again, same thing as I showed you, and at the beginning we picked up 10 mutations. This is technical where they are. It's almost not very important because we will do many more. Actually, the first set of mutations, those were 10. each of these mutations were found in a PXE patient, and we picked them up because it seems, according to the diagnostic statistics, that those are the most frequent mutations in the disease, in the patient's cohort. We saw the data. So we started with those. First, we set up an experimental system, and I'm not going into technical details because that's not very important. But what we can do, that we can generate the protein in a form when we can separately detect or analyze or study its transport function. Now you can address a question now that I'm always talking about a transport function, but we do not know what the protein is transporting, what is the metabolite. And this is true, and my talk is not about that problem, but even that we do not know what is the metabolite or protein transports, we can study its transport function, its transport activity in vitro in an experimental system when we use so-called artificial or model substrates, model compounds. And this plot shows that if the transport capacity of the normal protein, I mentioned you, we call it wild type in genetics, is 100%, most of the protein we studied are pretty good transporters. Not much worse, or sometimes we measured a little bit better, but it's really not a huge difference. And among the 10, we found only 2 where the transport activity was more or less gone. So these 2 mutants, we understand why they cause PXE. They cause PXE, but irrespective whether they go to the plasmamembrane or not, they cannot transport. They are inactive or very low transport activity, what they show. Here we have 7 other proteins which are perfect transporters in our in vitro system using an artificial substrate. So we do not understand why they cause disease. I have to mention you that there was 1 single mutant which we could not study in this system because it was not stable enough. That happens in science, you cannot foresee. But but in general, what we can tell you that basically randomly selected mutations, most of them are active. So here is the next question, what we address. Are they present in the plasma membrane at the place where their activity should be executed? You remember I told you that if they are not, they cannot do their job. So then we developed a next experimental system to study these proteins, whether they are localized at the same place at the Y type in the plasma membrane. It seems that this slide comes a little bit slowly. Yes, here we are. So usually people for this purpose use cell cultures, but we decided to use an in vivo experimental model, the mice or the mouse. I have to tell you at the beginning that this work, what I am going to talk to you now, is a very fruitful collaboration with Olivier Lesau's group, who works at the University of Hawaii, and he's also connected or a good friend of PXE International, not only good friend of our group. So we did this part of project together at both laboratories, and we designed these experiments to use the liver of living mouse to overexpress or to generate human proteins and to study human proteins. We did so because there is no closer organ or no closer biological system to human than the liver of a living mouse. It is much, much better. Our experiments showed that this is much, much better and gives much more reliable results than to use simply cell cultures. It's more work, it costs more money, but this pays back. So what we do, that we have the gene of the human protein, mutated or non-mutated. We had all of the mutations that separate in separate DNA form, and inject it on a way into the through the tail into the mice body, and it ends up in the liver. This is a so-called macro section of the liver, where each green dot shows one mouse liver cell which expresses the human protein. So this is so far so good. We found that roughly 10% of the mouse liver cells express our proteins, the human protein. So this is ready to investigate the localization. Here is again a microscopic image, which is a little bit more complicated than I showed you before, but basically the same thing. Blue is the nucleus of a liver cell, but this is not human now, but the mouse liver. Red shows mouse ABCC6, and green shows human ABCC6, a single cell. So one thing is that the human ABCC6 is in the plasma membrane. Great. This is what we expected, and this means, or this predicts, that this system is ready to study the localization of the PXE-associated mutants forms of the protein. And this is what I will show you on the next slide. I'm sorry, I went too far. Yes. Oops, I don't know why it wants to step twice. Give me a minute to go back. Yes, this is what I wanted to show you. Here, it looks like a complicated slide, but I will try to go through with you to understand everything. All of them are microscopic images, so this is one cell. We injected Y type, normal gene, and mutant genes into different mice through their tail. I also generated an artificial mutant, and this mutant, we knew that beforehand, will stay intracellular. So this is what we call in science as an absolute negative control. The normal protein, as I showed you before, goes into the plasma membrane. You see? Sometimes it's not that sharp, but most of the time it's very sharp, and it's clear it's in the plasma membrane. Here we have a mutant. This mutant looks very similar to the Y type, but this mutant was inactive in the transport assay. So we understood that this is a disease-causing mutation because it's inactive. Doesn't matter whether it goes into the plasma membrane or not, this is not functional, cannot transport anything. All the other proteins, I do not show all the 7 or whatever, I show you only 4 here for the sake of simplicity. All of them were localized different than Y type. You see that this is more inside than out in the plasma membrane. This mutant is completely in, this mutant is completely in, this mutant is completely in. Now, if we stop here, we can tell that, all right, we understand this is a disease-causing mutation because it's inactive as a transporter. This guy, this guy, this mutant, and this mutant were active as transporters, but they are not at the right place. So we now, one step ahead, we understand why they cause the disease. But can we fix this? Unfortunately, we cannot fix by the method I will show you, we cannot fix this inactive mutant, but the others, there is a hope that we can push them into the plasma membrane, and if they are in the plasma membrane as similar to the wild type, they may work okay. Let's see what can we do. At this point, we went and read many papers and figured out that there is a little molecule called 4-PBA. We refer to this little molecule, here is the structure, as PBA. So PBA is, on one hand, is an FDA approved drug. That's a good news if someone wants to use it in the clinic to do something with the PXE mutants. That will help a lot, that it's already approved for something else. Most importantly, we found a couple of papers when people use the very same compound for PBA to restore the cellular localization of another protein. This is a channel protein, and the mutations of this channel protein cause the disease called cystic fibrosis, which is completely different, and we are not discussing that disease at the moment. We just wanted to utilize the knowledge what other people at other universities or research groups gained using this compound, sometimes called as a chemical chaperone, sometimes called as a pharmacological restoring agent. So anyhow, they showed in cell cultures that they can push the mutant ABCC6 protein into the plasma membrane, just simply treating the cells with this compound. So we decided that we will treat our mice with this compound. And here are the results. I ask you to concentrate first on the left panel. I organized the images here that I always show you two different sessions of one protein. This is the wild type human ABCC6 injected into the liver of the mice, and it is present in the mouse liver cells. And it goes, as I showed you before, into the plasma membrane. If you add this drug, this compound, to the mice, nothing happens. Of course, nothing happens because everything was already okay with this protein. That was just a control to show that nothing wrong can happen. Let's see, here is a mutant. What would be an ideal target of this drug treatment, since the protein stays inside, you see? But unfortunately, when we added the compound, the drug, PBA, to the mice, did not help. Same thing with this mutant. If we do not treat, the human protein stays within the cells of the mouse liver. We give the mouse the drug, not too much happens. But look at this. This is the winner. This is what keeps us exciting, this mutant, and this is what gives a hope and promise. This mutant also localized within the cell, and that was the hypothetical reason why it triggers disease. But when we added the compound, the drug, to the mouse, the protein went into the plasma membrane, or at least a large portion of the protein was found in the plasma membrane. Somehow the drug, like in this case, worked on a way helping correcting the mislocalization of this particular mutant. So we wanted to test, or wanted to get another experimental system to reproduce the results, and then we went to cell culture. I don't want to give you again technical details, just look at this. The same mutant present in cell culture. These are dog cells. In vitro, that was in vivo. In vitro, we added the same drug, and instead of finding the protein within the cell, the drug helped the protein to go to the right place where hopefully it can do its job. So I want to stop here for a minute and make a little summary, because we are probably at the half of my talk. So we showed that abnormal cellular localization can be cause of the disease, and it turns out to be that most of the cases this is the situation. We also showed that with simple drug, which is already present in the clinic, an FDA-approved drug, the cellular localization can be corrected or restored in an in vitro experimental model, namely in mouse liver. So here we are now, and the next step would be, and then the next question I would ask that, all right, this is only localization. Can it be translated to function? Can you demonstrate in vivo in the mouse experimental system that correcting the cellular localization of a mutant accompanied with corrected function? And this is what I will show you soon, but if you have questions, we can discuss questions now for a minute or two or even longer, or it depends on you, we can wait until the end of the webinar when there will be room for more questions. So what should we...
We do have some questions, András?
Yes, yes, I'm ready.
Good. One question from Patrick is, what about the mutations that allow for partial function of the protein? Does it cause PXE in mice and also in people?
Actually, in principle, I don't know. We did not find yet. We did not find mutant which was half functional or 60% of with the function. My expectation or my idea is that a partially active protein will not cause disease, because unless the activity is very much reduced. And why I think so? I think so because in families where the parents are so-called carriers of the disease, what means that in one of the chromosomes they have a mutation and the other chromosome they have no mutation, they are not sick. They do not show symptoms, although they have in principle only half as much protein and half as much activity as somewhere in the normal population. So from that we can learn that 50% activity is enough to stay symptomless. But answering the question, we studied so far maybe 13, 14 mutants. I showed you only 10, and we could not see partial activity.
Thank you. Another question is, why has it been so difficult to identify the substrate that is being transported or not in by ABCC6?
So the trivial question would be that we are, or the scientific community is not smart enough. but let's take another option. Number one, that we cannot formulate. We formulated, not we. I say the research community, not only my group or not mainly my group. We formulated several hypotheses what it could be, but since we do not know enough about the calcification procedure in the skin, the so-called soft tissue calcification, we do not know exactly what should we look for. So all the hypotheses formulated so far failed. The other approach in modern science is a so-called hypothesis-free search for substrate. And that's complicated because then you deal with the whole metabolome. So metabolome means that huge amount of small molecules present in the circulation. The number is estimated to at least 1,000, but probably several thousands. And a biologically active compound or metabolite can be present relatively low level, a low concentration, and still be able to prevent calcification. So number one, we don't know what are we looking for. Number two, it's an extremely complicated chemical kingdom we are searching, and it's easily possible that what we want to find is present on a very, very low concentration, a low amount in the circulation. These are the major problems, but people, including my group, still working hard.
I'm glad you are. We have quite a few more questions. Are there other transporter types of drugs that could assist ABCC function in PXE?
Wait a minute. I did not talk about function yet. I just told you that the localization can be corrected. And I will show you how we try to figure out whether it can be translated. I showed you at the previous slide. Oh, sorry. How you remember that we need to develop methods, and I will show you in the rest of my seminar what type of methods we develop to demonstrate that the corrected localization is accompanied with corrected function. We are not here yet. I will end my seminar telling that I hope that in 6 months we will be answered this, or something like that, hopefully even sooner, we will answer this later question. Now, going back, how many little compounds are available? There are more than this. There are many. We simply cannot handle at this time more than one. Here we want to demonstrate that this is doable. And if we can demonstrate that this is doable, the number one good thing would be that we already have one compound which works, which is approved by the FDA. So it makes shorter, it results in a shorter time to start to use it in the clinic or to start clinical research. And if we can demonstrate, again I go back to the, if we can demonstrate that this compound works, that also shows that our experimental system is robust enough to check other compounds. And it's easily possible that other compounds which are around will work on different mutants of ABCC6 than the PBA, and we can open up a larger spectrum of mutations available for this idea. Does it answer your question?
I think it does, and I think that some of these questions now belong later on because they're
okay.
They're
would you mind that, because I just checked my watch and time flies, and I really want to show you a couple of other things, and maybe people who are interested will stay at the end and I can answer each questions or every question then. All right. Good. So I just want to remind you that we need to look for or develop a system which allows us to study calcification in mice and how the calcification is influenced if we express human ABCC6 in the mouse liver. And this is what follows now. Again, this is usual practice in research. We go and read and read and read, and found a beautiful technique published by a German group from Lübeck, and they used an artificial forced calcification of the heart of mouse. Again, I don't want to go into details, but what happens that with a metal device, which is extremely cold, we generate a cryo injury on the heart of a living mouse. And then what surprises me a lot, that the mouse doesn't care too much. All of them survive this little operation. And a few days later, when we sacrifice the mice, what we see, two different things. If the mouse was so-called normal, we see a little damage, a little tissue damage, but no calcification. Tissue damage, no calcification. If we use a so-called knockout mouse, the knockout mouse is exactly the same as this mice, but lacking ABCC6. So this is a genetic model of PXE that has been generated by Uni Vito's laboratory several years ago in Philadelphia, and then another mice variety, basically the same, has been generated in Amsterdam by Dr. Bergen's laboratory. So if we use this cryo injury on the heart of the PXE mice, which lacks ABCC6 from the mouse genome, we have strong calcification on day 3 or 4. You see, these white spots, white deposits are strong calcification. If we get the mice and stain it with calcium phosphate, that's the calcium deposit, normal, you cannot get any staining. If the mouse is a PXE mouse, you see very strong calcification at the surface of the heart in the muscle cells where we performed the cryo injury. So it seems that this is a functional test. This is relatively fast because it takes only 3-4 days, and it is ABCC dependent. If the mouse has ABCC6, no calcification. If the mouse has no ABCC6, calcification happens like in PXE. But I want to tell you, this is experimented. So in PXE, the heart never calcified. Of course, it's never cryo-injured. This is artificial, but it seems that we have the physiological symptom, calcification, and it's ABCC6 dependent. So it's a very good experimental setup to study calcification in vivo. And what we did, that we made, in science we always do time course and different experiments, and you can see that we did not measure at day 1, but at day 2 calcification was very low. We measure calcification like cutting out that piece of the, just cutting out this piece of the liver, dissolving calcium, and measure calcium on chemical reaction which generates color, and the color is proportional to the calcium content of the tissue. We have the normal mice, very little if any calcification. This is the background or the baseline, and we have 2 different knockout mouse models, and each calcifies, more calcification at day 3 than day 2, and a little bit more at day 4. So this is a time-dependent, ABCC6-dependent calcification. That looks like a good model. I just want to remind you what was the original question. If we insert the human ABCC6 protein into this mice, what calcifies so dramatically, can we prevent calcification? And this is what comes on the next slide. Here is the control, and that much calcification we measure. If we do not have ABCC6 in mouse liver, this is knockout. This is another knockout, but this knockout we injected with the human gene, and we show that the human protein is nicely expressed in the liver. Therefore, we have the green staining of the plasma membrane. And there is no red staining, as I showed you before, because this mice has no ABCC6 its own. And what happens if we do the cryo injury? We see very little calcification. And if we measure both here and here, this is very, very little comparing what we see here or here. This is strong calcification because there is no ABCC6 in the mouse liver, and this is little calcification since we injected the human protein. So the human protein provides a block of calcification, or it's called the rescue of calcification. And that reminds us to the physiological situation. If we have ABCC6 one way or another, here we restored it artificially, the calcification is much less than in the knockout. The knockout is the PXE mouse. So this is where we are now. This was, I don't know, I don't want to count how many years of work to set up in vitro and in vivo mice experimental systems. And what comes next? You can predict from the little pieces of the puzzle what should be done in the next few months. So number one, that we have a successful rescue of the fast and ABCC6 dependent calcification in vivo. That's very important, that we use an in vivo system. So this provides us a suitable method to investigate how can we fix the PXE mutations, not only the localization, but also its function. This is the plan what we are working on right now. This is, again, I have to mention you, this is in collaboration with Olivier Lesau's group in Hawaii. All the research goes parallel in both laboratories and goes very intensively. What we do, that we express again PXE mutants in mouse liver. I showed you that we are able to do that. We treat that mouse with the drug PBA. I showed you that this PBA is able to correct the mislocalization of the mutant, and we do believe that the mislocalization is the cause of the disease. In parallel with putting human protein into mouse liver, we induce calcification of the heart and wait a few days, and then we determine the level of calcification, and if we see that in a certain mutant the treatment with the drug lowers calcification, we can talk about rescue, and this is our research plan for the next couple of months. And I hope that when we are successful, hopefully before the end of the year, I will be able to send you a letter which PXE International can circulate, and we will be able to, first of all, we will be happy, open a couple of bottles of champagne, but we will have in hand a demonstration that this is doable and this is the right way to start clinical research. Now, if I have 5 more minutes, I want to show you another animal which is which gives us a beautiful tool to study the mutants. And this is zebrafish. Unfortunately, we cannot use this zebrafish. I would love, but it's not available. So we use this zebrafish, which is small and still very beautiful and very useful. The scientific name is Dario Rerio. Nice thing is that we know the genome as well as the human genome. We know the development of the fish. It's very, very fast, few days. We can knock down any genes by a very simple way. That's again technical, but it's very important because that's what we want to do. We want to switch off ABCC6 gene in the fish. And the good news is that together with the Univito group in Philadelphia, we identified the fish gene, which is very similar to the human gene by structure. So we address first the very trivial questions: what happens with the fish if we knock down this gene? That's again a few interesting images we go through together. This is the developmental scale of the fish, first day after fertilization, 3-day post, and 5-day post. We will see normal fish development, which ends up with a very nice, straight, young fish larvae, I would call, so early fish form, but very nicely developed. If we switch off the gene one way or another, we did it on two different ways because we wanted to be sure that we are on the right track. And even at day 3, we can see that something dramatic happened to the fish. The fish did not develop to give the normal straight shape, but it curves even more here, develops a kind of a structure which is not present in the normal fish, and it is even more pronounced after day 5, and after day 6 or 8, the fish in which ABCC6 gene is switched off, they die. So what we can see here, that there is a phenotype, there is a dramatic consequence if we switched off the gene in the fish. This is a developmental phenotype which is very different than in human. So the next question was that, okay, we learned something, but can it be applied, this knowledge or this experimental system or this organism, can it be used to characterize the human protein? I put here the question, what can we learn from the fish? As I told you before, the fish part of our work was in a collaboration with the Univito's group in Philadelphia. And the answer is yes. In spite of the very different condition or very different phenotype, very different consequence, what knocking down or missing the human gene or knocking down the fish gene, so they do completely different things. In human, it triggers a genetic disease, PXE, which is a moderately severe disease. It's not a good news, but it's not that strong as what you have seen in the fish. But in spite of this huge difference, we can use the fish system because we showed that if we inject the human gene, the human ABCC6 gene, into the knocked down fish, in the fish where its own gene does not work, we can rescue the phenotype. We can get the same normal development as the control. You see, so if these fish haven't got human gene, they would be completely crippled. injecting together with the chemical which switches off the fish gene selectively, if we inject that together with the human gene, this fatal consequence of knocking down the gene is gone, and we have an almost basically complete rescue. So this is a great system, because at least up to 90% this method works, and we can study the human protein and the mutant forms of the human protein in fish. Therefore, I put it here that in spite of the difference between fish and human ABCC6 gene function, the human gene can substitute for the fish one. Therefore, the fish can also be used, not only the mice, for characterizing PXE mutants. So we have two different animals. One is good for something, the other is good for something else. And I want to show you a table which summarizes the results so far we got. I don't want, this is complicated, and I have to tell you that this is only part of the summary of our results. But here, with the help of Yoni Vito's group, we injected all the mutants gene into fish and detected their capacity, whether they can restore the function if the fish gene has been knocked down. I showed you that the normal human gene and the normal human protein gives almost 100% rescue. These mutants, what we studied, and all of them are PXE causing mutants, give 0, 0.5, 1, 8, so relatively low or zero rescue. This is fantastic because this is what we expected, irrespective what is wrong. Functionally, we proven that these are real mutants. There is only one mutation which gave us partial rescue. And if we go back to this mutant was at the right place. But this mutant was, I told you, inactive. Inactive in our hands means that it has less than 10% activity. We cannot measure, our method is not sensitive enough to measure below 10% activity of the normal. If this mutant is 5% active and we injected a huge amount of the human gene, it can provide some function. But the take-home message is that in fish we can study the functional characteristics of the mutants, what we already analyzed in mice, and we can study their function, although the function on the level of the organism is different in human and in fish. Science goes sometimes on a tricky itinerary, but we try to learn as much as possible from our wonderful animal models. And basically, I just want to give you, to summarize or to repeat what we went through, two examples. I told you that we want to know what are the consequences of a given mutation amino acid replacement in a PXE condition. I go back to the transport panel I showed you half an hour ago, and I picked up this mutant. This mutant we found inactive or very little activity, if any. There is no difficulty to understand why it is disease causing, because it has very low activity. It goes to the right place, but even at the right place it cannot do too much of its own job. Here is another mutant. This mutant, again a disease causing mutation, pretty much active, almost, or I would say it's as active as normal. This high activity will not explain us why was it disease causing. We went to the mouse liver and we found it within the cell. It did not go to the plasma membrane. So if it's active within the cell, does not help. It should be active in the plasma membrane. So we understand why this mutant is disease-causing. However, that was the major aim or the major dream before we started this project, that we could restore its localization back into the plasma membrane using a relatively simple drug, PBA. So here we are, and we are trying to find other mutants behaving like that. And as I showed you a couple of slides ago, we want to see whether this better or corrected localization can be translated to anti-calcification function. So one of the very last slides of the presentation addresses the question that what does our research mean to the PXE community, because that's the most important thing. I think that we are halfway ahead to answer the question whether is it feasible to correct certain PXE mutations, more precisely the consequences within the cell of certain PXE mutations, at least in animal experiments. And we have to use here mostly the mouse system. But also at the background we have the zebrafish. If we can correct the function with the methods I showed you, the cryo injury, the tail injection of the human gene, and so on and so on. So it's a little bit complicated, I know, therefore it took us years to put this together. But if we will be able to show that, that would support a future clinical research which can be initiated hopefully somewhere, showing the same ideas, same drugs or similar drugs. And that's what we hope. So I want to give you a list, what I will not read it now, but there are several postdocs and graduate students involved in this research in the past couple of years in my group. We got very nice partners for fruitful discussions. I mentioned you Olivier's group and Yoni Vito's group as very successful and fruitful collaborations. For research we need money, and I have money from Hungarian sources, but also from US and also from PXE International, and I cannot be thankful enough. And we cannot be thankful enough to our mice. They discuss what's going on, and we discuss what's going on. And in the background, those are also mice, but next time I will show that those with little tanks for the zebrafish. And this is why I am ready to answer your questions. And this is some announcement from PXE International. And thank you very much being with me, and looking forward to your questions.
Thank you so much, András. Though you made that very accessible, some complex research. I'm going to sort of jump to the very end with a question, and people really want to know, you know, if 4-PBA can rescue calcification, when can we get that? When can humans get that? Or if we could have injections of ABCC6 gene into our bodies, could it reduce calcification like it did in the mouse heart? So can you address kind of the timeline and what needs to happen before? I know you already started.
No. So this is two completely different. I mean, the idea is very logical and shows to the same direction, but from the point of view of science and practice, these are two completely separate issues. And we deal with only one of them. We deal with the problem when someone, a patient, has a mutated gene. And the mutated gene can give birth to a full protein, but the protein does not go to the right place within the liver cell. So this is the basic starting point. We sort human mutations and try to classify, and we will deal with those which are not at the right place within the cell. Otherwise, they are capable to show transport activity. Now, these are the candidates, or these are the targets of our research. And just to put it in a precise context, the drug what we use as a potential treatment, or at least treatment of the mice, not preventing calcification directly, it helps the protein to go to the right place. It helps it makes the protein to be at the correct physiologically relevant place in the plasma membrane, and then the protein can start to work in the plasma membrane, and the protein, the corrected localization protein, hopefully will prevent calcification. Just tell me, was it one part of the question, but was it understandable?
Yes, I think so.
Okay. You asked about injecting, or I would not call injecting, but somehow providing the gene to the PXE patients. And that's what is called gene therapy. And first of all, I have to tell you, I am not a physician, and what I do here is mostly biology. So once, hopefully, the gene therapy will be in principle available in PXE. I cannot comment that because I'm not an expert. Gene therapy is a big hope for many other diseases, not only in PXE, but it's very, very difficult, and at the moment it's not 100% safe to perform. If you read the literature, there are many, many trials, but it's not routine at all. So I cannot predict when a gene therapy will be available in PXE. I am afraid not in the near future because of the safety issues. So this is my answer to that. Again, I want to tell that I'm not an expert in gene therapy.
And gene therapy, is that being used in any genetic disease that you know of?
Yes. As I told you, I teach human genetics, molecular genetics, so I teach gene therapy to my medical students. And gene therapy has been used in several very, very severe, I would call fatal genetic diseases. Fortunately, PXE is far from that. And all the problems came out on those diseases. All the problems, so how can I explain to you? The major problem is that you have to deliver the gene into the human cell from where the gene is missing. And a good gene delivery means that the new gene, the human gene, or in this case, let's suppose ABCC6, stays forever in that cell. Otherwise, it will have only for a few weeks. So to use good vehicles, good methods to get the gene permanently into that cell, people have to use viruses. Viruses insert the gene into the normal genome of the host cell, and it will be there forever. But that is the source of the major problems. If the gene goes into a wrong place, it can do horrible problems. It can trigger leukemia, what happened in a big trial about 8-10 years ago, and that slowed down the gene therapy using that kind of viral, the best, I would say, the best viral method. So gene therapy, therefore I tell that gene therapy is fantastic. People invest lots of work, and I do believe that once all the safety problems will be solved or will be put down to an acceptable level, but right now this is the major problem, that you cannot predict what else happens. If a virus enters into the cell, a virus carries the therapeutic gene but can make side effects which could be dramatically bad. And that happened. It's not only theory. That happened in a large study in France with several kids, and more than one kid of the trial developed childhood leukemia.
Okay. Do you have time for a couple more questions?
Oh, yes. I have to leave 45 minutes from now.
Okay.
For another program.
I'll try to make this one brief. I'm wondering if one of our members is wondering if you know the reason on a molecular level why some cells responded to the added PBA drug and some cells did not respond.
You mean this? Probably what you wanted to ask? It's a very, very clear question. Why some of the mutants responded and others do not?
Yes.
And we have some idea, and this is a side direction of our research. I will explain you in a minute. So, in general, a membrane protein, like each protein, is manufactured somewhere around the cell nucleus. I try to go back, okay, and try to just use one of my pictures. Give me a minute, I find it. So each protein is manufactured somewhere close to the nucleus. And a plasma membrane protein, I need to go back two more. Come on. Yes. Give me a, yes, probably even more. It's always slower than, yeah, or even one more step back. I expected to change slides a little bit faster, but I don't know why it does not go faster. My computer gets tired. Just give me one more minute, because that is a very good place where, yes. So here is a cell, a typical cell. This is a liver cell. Here is the nucleus. And somewhere very close to the nucleus, all the proteins, there are, I don't know, I guess 10,000 different proteins or 6,000 different proteins in a liver cell. All of them are manufactured somewhere here. And by a very complex trafficking itinerary, they go into the plasma membrane. There are several steps. And you can imagine that each step is like crossing a border. Like when you go from the US to Canada, there is a border guard. And I don't know whether it's true or not. I guess that they, in principle, can stop you and check your passport. And if you have no good passport, will not allow you to go to Canada. Probably it's a very stupid example.
No.
Okay, so there are checkpoints. There are, let's suppose, 4 different checkpoints. At each point, a system, a complex, active system, checks the protein and tells that, okay, you guys look okay. You look like normal. You can go. Here is another one. Still go. You can go. Here another one. You can go. Finally, it ends up into the plasma membrane, because it had some kind of address that it should go into the plasma membrane. Now let's suppose what happens with a protein which is a little bit different than the normal because of that mutation. And let's suppose it's a little bit crippled. Then at one of the checkpoints, the border guard says that no, we don't want you. You are not good enough. And it goes to the trash and cannot reach plasma membrane. And that's what we have seen with our mutants. They accumulate inside, and sooner or later they go into the trash. So they are eliminated because we need space for other normal proteins.
And your research was able to, the ones that were successful with PBA were able to move
interfering.
The membrane.
Exactly. So the PBA, according to the research, and part of our research goes how PBA works. So the PBA basically somehow helps the little bit crippled protein to cross the first border. And most of the protein, some of the protein stops at the first border. If someone helps it to cross the first one, we'll be able to cross all of them. Some of them, and some of them is stopped at the second or the third border where PBA cannot have. We, at the beginning, or when we start to work, we cannot tell what is wrong with this protein, whether it was hooked at the third checkpoint or the first checkpoint. If it was hooked at the first checkpoint, PBA may help. And if I go again further, this protein, we knew from different other experiments that indeed it was most probably hooked up at the first checkpoint. And PBA helped to cross that, and from that point the protein looked okay and went to the plasma membrane, here and here too. So therefore it is not predictable exactly or not predictable at all whether a mutant will be responsive to PBA treatment like this one or not like this one. But on the pipeline in drug development, there are different drugs. And maybe another drug, which we never tried yet, but we want to try that in the future, maybe another drug helps to cross the second checkpoint or the third checkpoint, and we will have more possibilities to correct cellular localization and hopefully function. Did it answer your question?
Yes.
Okay.
Again, you make a very complex topic.
Yeah, this is the chance. Therefore, we have to analyze many mutants and go and pick up those and those and those and those. But at least I strongly hope that at the end we will be able to demonstrate that this is doable. Then we go for other compounds.
And one last question from a member. Is the speed at which you can conduct your work, is that related to the level of funding that you receive?
Ooh, of course, but it's not that simple. Let's suppose that I do not buy lottery tickets in principle, but let's suppose I bought today and on Friday I got $1 million. I could not accelerate immediately my research because I need a very well-trained staff, and it takes several months, if not years, to be able to do all of the tricks what we developed. So without funding, I cannot do that. With too little money, I cannot do that. If someone gives me 10 times more money, next week I cannot do 10 times more research. I don't know whether...
But you could ramp up. No, that's a very good answer to this question. It's complex work and it takes you some time. It's
complex work, and definitely more money on a longer term. We'll be able to study more mutants at the same time. For instance, right now the major cost is to keep the mice. We deal with here each experiment, what I show you here, behind this 10 mice, behind this probably 15 mice. So just to get 2 points on the on a plot is 25 mice. Generally we use 20-25 mice each 2 weeks. So it's hundreds of mice a month, and that costs a lot of money. These are kept in special animal facilities under sterile conditions and so on, needs some special care. So this is a major cost. Having more money, probably we can train more people, and after a couple of months, maybe half a year, we can double the number of mice or triplicate the number of mice and triplicate the flow through, means the number of mutants we can analyze.
Okay. Well, with that, I think we have answered most of our questions, and I want to thank you very much for your time, András, for being with us, and we hope to be able to continue sending you money, lots of money, in the future for PXE International.
I have to tell you that I never made a webinar, and I enjoyed each minute. Not because I enjoyed not because I enjoyed my explanations, but somehow I felt that there are people on the other end of the wire or the line who are interested, and that makes us very, it gives a very good feeling.
That's very good, and they're very interested in you. I have a comment from one that we really like you, Dr. Váradi, and keep going. So thank you again. We really appreciate it.